We discuss our immune system has the two types of immunity, the innate immunity and the acquired immunity. And the innate immunity act as a non-specifically against all the pathogens, whereas the acquired immunity is specific and one cell or molecule acts against the only one pathogen. And the acquired immunity comprises the humoral immunity and the cell-mediated immunity. And this humoral immunity acts against the pathogens that haven't entered any self cells, and the cell-mediated immunity act against the pathogen that have entered some self cells. And these are the lymphocytes that play a key role in the humoral as well as the cell-mediated immunity. And the lymphocytes are formed from the stem cells in the bone marrow. And the lymphocyte differentiated into the two types, B lymphocytes and the T lymphocytes. B lymphocytes are produced in the bone marrow itself, and the T lymphocytes are processed in the thymus. And the B lymphocytes are the mostly responsible for the humoral immunity. The cells responsible for the cell mediated immunity are the T lymphocytes. And some of the types of the T lymphocytes are the cytotoxic or the killer T cells, helper T cells, suppressor T cells, and the memory T cells. And each T cells possesses a number of receptors for a particular antigens, and the T cell receptor is in the many ways similar to that of the antibodies, and it can recognize a specific antigens. And the TCR is a protein made up of the alpha chain and the beta chain joined by a disulfide bond, and each chain, alpha and beta of the TCR, has the two extracellular domains and a transmembrane region and a short cytoplasmic tail. Here with the diagram, we can see the short cytoplasmic tail, and this is T receptor, and the alpha and beta chains are here. And the alpha chain has got two extracellular domains, alpha 1 and alpha 2, whereas the beta chain has got two extracellular domains, beta 1 and beta 2. The alpha 1 and the beta 1 domains are on the periphery, and the alpha 1 domain and the beta 1 domain are similar to the variable region of that of the antibodies, and these are also known as the V alpha and the variable beta domains also. The alpha 2 and the beta 2 domains are similar to the constant region of the antibodies. Like the antibodies, TCR can recognize and bind antigens. Then the antigen recognition is the function of the variable region, and the antigen binding site or cleft is formed by the alpha 1 and the beta 1 domain. Here we can see the alpha 1 and the beta 1 domain is forming the antigen binding site, and these are the alpha 1 and alpha 2 domains, beta 1 and beta 2 domains. There is the transmembrane region, these are the constant beta regions, and the variable regions are to the periphery. And there is a diversity of these TCR, and the T cell receptors have a diversity similar to the antibody diversity. In the antibody diversity, we have seen there is a recombination between the VDJ recombination and other types of gene recombination take place, and to generate the millions of the antibodies, such like the TCR diversity is also there, which also arises from the gene rearrangement. In this, the gene for the variable regions of alpha chain V alpha is constructed from one of the 70 variable segments and one of the 61 and J segments, and thousands of different combinations are possible. And the gene for the variable region of the beta chain, variable beta, is constructed from one of the 52 variable segments and one of the two D segments and one of the 13 J segments, and thousands of different combinations are also possible in this. And when an alpha chain combined with a beta chain, millions of combinations are possible. A given T cells has one combination and is specific for one particular antigen.